Perhaps I can begin just simply by describing vaccine development. I mean, at its heart, vaccine development is a relatively simple concept. First, you prove that the vaccine works, is safe and effective. You make the vaccine, and then you use the vaccine. And usually we have 5 to 10 years to actually prove that the vaccine works, to make it, develop it, and do all these things. That comes at a cost of about $1 billion and has a 93% failure rate. But under the pressure of this pandemic, with 8 million infections and 400,000 deaths, we're being asked to speed the process up and to do this work that normally takes 5 to 10 years in 12 to 18 months. So to cut to the bottom line up front, can we prove that a vaccine works? Probably. It's likely. Can we do it in 12 to 18 months? That'll be tougher, but if everything works, possibly yes. Can we make it in sufficient quantity, with sufficient quality, and at an affordable cost? That's a tougher one. And finally, will we be able to use it with equity and access for all? And that's possibly the toughest one of all, but that's the one. That is the question that will bring us to the end, which is a significant reduction in COVID-19 burden and disease that will result in alleviation of the suffering that this pandemic has brought to the world.
Thank you for that, Dr. Kim. And our audience will jump in with questions soon, I'm sure, but I have one for you to kick this off. You worked on the HIV vaccine, and obviously that has been a multi-decade effort, and we still don't have one. What gives us confidence that it will be different with this particular coronavirus?
That is a great question. And there are a couple of reasons why I think that we will probably be more successful and sooner with the coronavirus vaccine. The first is actually something that we don't have the full answer to. Still a largely unanswered question, but I think we probably are gaining enough information to make a reasonable response. And that is, with a typical disease, say measles or hepatitis A, after you acquire the infection, everyone has a slightly different course. But in the end, your body's defense system, the immune response, is capable of controlling the virus and eventually eliminating it from the body. And those same protective responses do not allow the virus to come back. So you have immunity or protection from reinfection. That is not the case with HIV. With HIV, and to some extent with TB, an initial infection can be followed by reinfection. And that reinfection really indicates that the immune responses that are developed normally in the normal human body to infection are insufficient to get rid of the disease, but even more important, to protect against reinfection. With COVID, I think there's increasing evidence that people who have been infected are, at least for a period of time, protected against reinfection. And that's actually a very critical concept. The second part is, and it comes from animal models. We have, or we often say as vaccine developers, that mice lie, monkeys exaggerate, and only humans tell the truth. But really, the monkey model is the closest thing that we have to a proof of concept before we actually test the vaccine in humans. And now there have been 3 reports of tests using the current human vaccines against COVID-19 protecting monkeys against challenge. And so that would mean that at least in a non-human primate system, which is what monkeys are, vaccination can protect against infection. And that's actually a very important proof of concept. So next on to people.
So let's break that down a little bit. There seem to be hundreds of vaccine candidates. What are the approaches? But they all sort of take different approaches. What are the main approaches we're taking with these vaccines?
Okay. There are 2 that were very fast. We call them nucleic acid vaccines, and they are RNA and DNA. So you remember from biology that when you start with a gene, which is DNA, in order to get that gene made into something, you actually have to use something called RNA. So it goes DNA to RNA, and the RNA is then translated into protein, which then becomes the material from which we can either create new parts of the body or create other structures in the cell, or make things that will develop into those structures. And so the two vaccines that were started first were DNA and RNA. And actually, within a few days of the sequence of COVID-19 being published, companies that make DNA and RNA vaccines are reported to have developed the concept for the vaccine. And within a week, they had made the vaccine. And shortly thereafter, they had injected it into animals. And so the first of these, roughly 8 weeks after the initial announcement of the sequence of the virus, entered testing in humans, and that was the RNA vaccine from Moderna. So those tend to be fairly quick, DNA and RNA, and actually in April, the test of the DNA vaccine made by Inovio in humans started as well. So again, a very fast development process. The next set of vaccines that were out there were what we call vectored vaccines. These are little pieces of the genetic information of the COVID virus inserted into another virus. And one of the more commonly used ones is a common cold virus called adenovirus. And the adenovirus type 5 is the basis for the Chinese CanSino vaccine. A chimpanzee adenovirus is the basis for Oxford's vaccine, which is now being taken over by AstraZeneca. And an ad type 26 vaccine is the vector for the vaccine that Janssen is making, which is a part of Johnson & Johnson. The next set of vaccines are conceptually easier to understand. These are what we call whole inactivated virus vaccines. And in a whole inactivated virus vaccine, you introduce the virus into cells. The virus grows as it normally would. You harvest the virus from the culture medium, and then you purify it and kill it with heat or formalin or other chemical substances. And those whole inactivated virus vaccines, are relatively easy to make. They're relatively cheap, and we have examples of them. The inactivated poliovirus vaccine, for instance, is a whole inactivated vaccine. So each of the concepts has strengths. Some of the concepts are faster than others, but really, we have a host of proteins, of vaccines now entering the second stage of testing in humans, which is incredibly fast.
And great news as well. Well, I believe we have some questions from our audience. I'll take the first one, if we can fade that in on the screen below. Stand by for a second. I know the questions are out there. It's just a matter of getting it on screen. Oh, looks like we're having some technical difficulties, but we're back. Well, I guess I will ask another question. Oh, here it is. I guess we're not going to be able to fade it in on screen. It's a question from Kevin from our community. What do you think the effective rate of the first vaccine will be?
Almost impossible for me to tell. We're targeting something above 60 to 70%. And the reason it's difficult to tell is there hasn't been much data put out yet. I mean, we've advanced to phase 2. We've seen some of the data for protection in monkeys, but we've seen relatively very little data in humans. We heard Moderna report on 8 out of 45 of the people in their phase 1 trial. We've started to see some data from CanSino, which is an Ad5 based vaccine. But it's actually been difficult to form an opinion because there actually hasn't at this point been a lot of data published based on the human trials. We're starting to see some small animal data, from Inovio, which is a DNA vaccine, and from Moderna, small animal data, which is an RNA vaccine. But again, this makes it a little difficult to prognosticate. But I would guess that you're going to need to see a fairly significant effect And actually, it's not only the actual number, but how reliable that estimate is. And that's the thing that the US FDA, the European equivalent of the FDA, and the WHO have been pushing, that we really need enough precision around the estimate to really make a clear statement that this vaccine actually works and is safe.
Yes. In the meantime, wear masks. All right. The next question comes from Frank Hennessy, and you can see it there at the bottom of the screen, so the technical difficulties have dissipated. COVID-19 seems to affect different people in different ways. How do you select a vaccine that is effective for everyone?
So that's actually another great question. But if you look at different diseases affect people in different ways. COVID is actually very interesting because 80% of people are either asymptomatic or have mild symptoms. 14 or 15% of people have significant symptoms. 5% of people get very sick, and 2 to 3% of people die. That's actually, a pretty reasonable distribution of illnesses. And I mean, measles is the same. Some people have very mild symptoms, have the rash. Other people get much sicker, and some people actually have severe complications from measles. And in places like Madagascar or in the Democratic Republic of Congo, children die of the measles. So again, but a single measles vaccine protects us all. And I think that's one of the other things about vaccines. When we think about treatments and antibiotics, resistance develops fairly quickly to an antibiotic. But we've had measles vaccine for decades, and there isn't resistance to it. So again, resistance develops more slowly to vaccines. Now, the one exception might be influenza vaccine. And we don't know whether COVID is going to go into influenza-like mode or not. And that's one of the things that we'll know more about in a year or 2 years or 3 years as this epidemic matures. But by and large, you develop a vaccine against measles, mumps, rubella, hepatitis A, hepatitis B, and that vaccine will work for a very long time.
One of the greatest public health interventions in world history. So our next question comes from Laura Pearson. My biggest concern with vaccines is side effects. Is it possible to test the long-term side effects in the short term? And if so, how?
So when you're developing vaccines, one of the critical features is efficacy, whether the vaccine actually protects against infection and disease. The other critical parameter is safety. Most of the side effects that we can look at in standard clinical trials occur within the first month to 3 months occur immediately, or in the first 1 to 3 months after the vaccination. The longer term side effects actually require much longer periods of time and sometimes much larger populations in order to see the effect. So governments, regulatory agencies like the FDA or EMA have set up systems to provide surveillance for adverse events, to pick up rarer adverse events. And so that's a very important question. But given the number of people that will be involved in the testing, we should be able to detect the relatively short to mid-term effects, so out to roughly a year or so. At relatively low levels, so under 1%. So hopefully, we will continue to collect safety data for the period of observation, which is for clinical trial typically a year to 2 years. We will continue to collect information because these vaccines in particular are being pushed towards efficacy. And I think many of us understand that as vaccine developers, the other and very critical part of what we have to do is ensure that we have vaccines that are safe. So, but a great question.
Yes, and I wonder if this will be a vaccine where you need to get it every year, like influenza, or you require a booster at some point, or because that will also affect my real question, which is about kind of manufacturing and distribution. Obviously, if it's one shot and done, then maybe you need just 7 billion doses. If it requires a booster, we're starting to get into some really crazy numbers. How are we going to manufacture and distribute these vaccines, Dr. Kim?
Yeah, you notice I left that part of the question blank. And the reason is, so normally when you're doing vaccine development, the last few years of your 5 to 10 year cycle, you're planning the manufacture because you're already in phase three. And if you're a vaccine company, you don't actually make any money on a vaccine until it's sold. So in the final year, 2 years of testing, you're in the process of making sure that you can have enough available early on so that immediately after approval is given, you can move to sales. That would happen in 5 to 10 years. Now we have 12 to 18 months. And where do you start planning the manufacture? So Operation Warp Speed, which is the US government program, is already saying that they may actually make vaccine at risk. And what does that mean? It means that while we're beginning the testing, the government will be manufacturing doses of vaccine at risk, not knowing whether the vaccine works, but just so that those vaccines that have entered the final stage of testing, when and if they're shown to be safe and effective, there will be doses of vaccine available for use. And that's actually a remarkable thing. And it happens, and it can happen. It doesn't normally happen, but it can happen this time because the US government is underwriting the risk for the manufacture of vaccine. So if you were a company, you wouldn't do things completely at risk. You've usually gone back. You're pretty sure that what you have in phase three is going to work because you don't want to waste time and money on a vaccine that isn't going to be safe or effective. What the US government is saying is, look, this is an emergency. And because it's an emergency, we are willing to carry some of the risk. And that allows the company then to start manufacturing before they actually know. Other organizations are planning a similar strategy. So as you're rushing to do what we call the phase three trials, the tests of efficacy and safety, at the same time, companies are going around the world to identify partners because they know that an individual company might not be able to make 1 billion doses, but if they partner with people in South America and India and Korea or Japan, they might be able to put together enough manufacturers to make enough vaccines so that over time we'll be able to produce the 7 billion doses that are required. If we only need one shot, if we need two shots, that could be 14 billion, as you pointed out. And if we need a late booster, that would be even additional billions. But the problem of the duration, or what we call the durability of those protective immune responses, really we won't know until we look at the curves of decay and we understand what the protective immune response is, something we call a correlate of protection. And those are actually going to be really important because once you have a correlate of protection, you no longer have to do a phase three trial in 30,000 people. You can do a much smaller trial much faster and really look to see whether a new vaccine or a modification of the vaccine, which may be better, develops that particular lab test, what we call the correlate of protection. And that really shortens development time and decreases the cost. And so one of the goals of the activity that's going on now is both to show safety and efficacy, but also to try to identify what we call the critical correlate of protection.
So even with that kind of development of these breweries for vaccines at risk or repurposing for COVID vaccines, it seems like we're not going to have 7 billion doses kind of ready to go, 12 to 18 months from now. How are we, or what in your opinion is the best way to prioritize, who gets the first doses and where and all that kind of decision making?
So, I think that countries will, countries, particularly those countries that have put significant amounts of funding towards, accelerating the development and manufacture of vaccines, are going to want vaccines to protect their own populations first. There have been commitments. For instance, the President Xi of China has said that vaccines are a global public good and that they would share those vaccines with other people. The question is how quickly. And on the other side, though, we have an organization called the Coalition for Epidemic Preparedness Innovations, or CEPI. And CEPI is a group of now what was initially 13, now probably 15 or so countries, plus the Gates Foundation and the Wellcome Trust and other charitable funders, the European Union, for instance, and they put together a $2 billion portfolio that allows them to develop vaccines very rapidly. they've funded many of the companies that are now moving vaccines into phase 2. And that's important because CEPI makes a company sign an agreement that it will provide global access for the vaccine, which is to provide adequate supply at a reasonable cost. And the CEPI group, there's a secretariat and countries and people who sit on their board of directors who will help to discuss the appropriate allocation. And we have to also consider organizations like the World Health Organization or the United Nations or other organizations that actually now should be talking about what we will do when and if we have a vaccine that's safe and effective. Because I think as you were kind of hinting, the time to make that choice is probably not when you have the first vaccine that works. Because when that happens, there will be a huge pressure to vaccinate us first, or them first, or this group first, or that group first. And really, if you haven't made your choices in advance and you can't defend it in a transparent and principled way, there's going to be a lot of recrimination and perhaps some unnecessary death as a result of that, or unnecessary infections and deaths as a result of that. So we need to start working now. We need to get together to talk about this.
Well, it certainly doesn't help that the U.S. is no longer participating with the WHO or CEPI. And it seems to me that healthcare workers and the elderly, who seem to be most at risk, should be maybe the first candidates. But that's just my opinion. I'll be interested to see. There's another audience question, I believe. Let's go to that if we can. Here we go. When there are multiple vaccines on the market in 2 to 3 years with varying efficacy rates and presume no safety signals, how does the world prevent the allocation of better vaccines to the rich and lesser vaccines to the poor?
That's a very interesting question. And when we have one vaccine, I'll be happy. When we have 3 vaccines, I'll be really, really happy. You know, we have to make, I think, as you pointed out, maybe 14 billion doses. That's a lot of vaccine. I mean, when you look at annual production of vaccines, that is tremendous. So different countries in the world will make different vaccines. That happens currently. And there will be some standards. The WHO has already developed what we call a target product profile for vaccines that it will review and hopefully approve very quickly for or recommend for use in countries around the world. And so hopefully all of those vaccines will pass the minimum requirements that are set both by the agreements that were put together by the regulatory bodies, the WHO, the US FDA, the EMA, all the different vaccine regulatory bodies in the world came together and put together a document. And hopefully then there won't be as much variation in what we would consider to be a minimal level of effective vaccines.